mutations in a hospital setting. I think that's right, right?
Perfect.
All right, excellent.
Thank you so much.
Evan's a famous guy, you know? You know, how many people know that? A few of us.
Don't believe everything you read,
if it wasn't for Evan, that clinical center over there, you know, the NIH, might not be the same as it is now. So maybe you'll tell us why at some point along.
I will. I'll tell you about a lot of collaborative work. So it definitely is not a personal glory story. It's a lot of different people with different backgrounds coming together.
But it's a very important story.
Well, we'll see what you think. I'll let you all be the judge.
Okie doke.
But you know, for the students here, I figured I'd give you kind of like an introduction to where I came from. So I got my PhD in bioinformatics, like you all are doing, and I did very much non-clinical work. So I was working with Charles DeLisi. You can ask Dr. Athey all about him and all the wild stories. With him, I was doing a lot of function prediction, interaction network, etc. I also worked with Daniel Segre at Boston University and did flux balance model analysis and understanding genetic interaction network, genetic perturbations impacting metabolism. So pretty like theoretical, basic research type bioinformatics. And looking for a postdoc, I was really interested in applying my bioinformatics skills to more clinically relevant research, and that brought me to NIH in the lab with Julie Segre. And I was really fortunate to become linked up with clinical microbiologists and clinical epidemiologists at the NIH Clinical Center. And we were involved in these broad collaborations with the goal being to apply whole genome sequencing to study the transmission and evolution of bacteria that are responsible for hospital-acquired infections. That's kind of the jumping off point, and I'll tell you a little about some of the work that I've been doing. So, no signal. Come back in. Maybe it didn't like going to sleep. There we go.
Imagine how important a problem this is. You know, just the C. diff infections alone at the University of Michigan.
Yeah. Yeah, that's the big organism here a lot of people are working on. But I think the overall numbers are pretty shocking. So I was pretty naive to the problem when I came to NIH, and here are like some of the base numbers. So complicate 5% of all hospital admissions, like 2 million people in the US alone. And I think obviously the big number, it results in roughly 100,000 deaths each year just in the United States. And you know, on first glance, these are pretty shocking numbers that in a Western culture where antibiotics are plentiful, that infectious disease should exact such a toll in our healthcare system. And ultimately, I came to learn that there's two primary reasons leading to these really high mortality rates. And the first is in a sense a good thing, which is that we're much better at practicing medicine than we used to be. And advances in things like chemotherapy and stem cell transplantation are keeping patients who otherwise would have succumbed to their illness alive. The flip side of that is these treatments often leave them in a very immunocompromised state where they're susceptible to infections that normal healthy people aren't susceptible to. And the second major driver has been a really alarming rise in antibiotic resistance of these infections over the past few decades. So a second thing that I learned upon studying hospital infections is that not all hospital infections are staph infections, despite what you see on hospital dramas on TV. And in fact, while gram-positive infections like Staph aureus are important, it turns out that 2/3 of the deaths of hospital-acquired infections are from gram-negatives. And here's some of the major gram-negative players and their relative abundances in ICUs across the United States. And you know, they're already a cause for alarm because of the mortality rates, and they're becoming an increasing cause for an alarm because some of the major players have shown a really striking capacity to evolve resistance to antibiotics over short periods of time. So this is data from the same study looking at gram-negatives in ICUs across the United States. And now what I'm showing you is the rates of multi-drug resistance in 1993 and 2004 for all these different species. And you see that for some of them, just in this roughly 10-year period, you're seeing huge increases in multi-drug resistance. So the two organisms I'm going to talk about today are Klebsiella pneumoniae and Acinetobacter baumannii, where you see a 3-fold and 6-fold increase in multi-drug resistance in a 10-year period. And the story is actually even worse than this, where there's strains of Acinetobacter and Klebsiella that are now being seen in ICUs in the United States that are resistant to every single antibiotic we have. And we've seen some of these strains in our hospitals, and essentially when a patient has an infection with this sort of organism, there's nothing you can do for them. They're put on palliative care, and that's all that can be done. So it's really gotten to the point of being a crisis. And fortunately, the powers that be are now starting to recognize the severity of this problem. And just in the past year, the CDC and the Presidential Commission of Science and Technology have released reports kind of laying out the path forward for how to deal with this crisis. And I just kind of want to give you a quick couple of bullet points of the highlights of these studies, because I think they pretty much are saying the same thing. And the first thing they're both saying is that we need more effective prevention strategies. We're at a point now where we have untreatable infections in our hospital, and what needs to be done is a better understanding of how infections spread so that we can implement more targeted infection control procedures to try to get a hold of them. Second, we need more informed treatment strategies. So when the doctor has a patient with an infection in the hospital, it takes at least 48 hours for that doctor to know with certainty what's causing the infection and what drugs that infection is susceptible to. So for that first 48 hours, the doctor is essentially treating the patient blindly, which is clearly not optimal for treating the patient effectively, but it's equally bad in terms of giving drugs that could potentially lead to the evolution of resistance. So we need technological breakthroughs that will provide doctors with information to ideally treat patients in real time. And finally, to state the obvious, we need more drugs. And the current pipeline, especially for gram negatives, is dry. There is nothing in the pipeline. And the flip side of that, in addition to developing more drugs, we need to better understand how to use the drugs we have to limit the evolution of resistance and maintain their efficacy as long as we possibly can. So yes?
Did you want to add a bullet down there, Evan, about agricultural?
Yeah, yeah. So that was in both reports, and I think that kind of goes to stewardship, like how we as a society and at a global society are disseminating and using antibiotics, and how they should be regulated both within a single healthcare institution and across the world. And it's clearly a global problem where resistance evolving in one part of the world or in one institution affects everyone. And those are the sort of solutions that are required. So what I'm going to talk about is a potential genomic revolution in hospital infection control. And I'm going to do that in the most egotistical way possible by just telling you about my work, but there's been a lot of really cool work in the past couple of years that I think has recognized that genomic can really be a game changer and something that can immediately be translational in this realm. So I actually wanted to first quickly mention two projects that I worked on that I think are maybe more interesting from a computational perspective and a comparative genomics perspective. But then I'm going to spend the bulk of the talk talking about two problems I think are more clinically interesting, just to kind of give you an idea of the seriousness of the problem and how genomics might really impact these serious problems. So the first project I worked on when I came to the NIH was using whole genome sequencing to understand an outbreak of multi-drug resistant Acinetobacter. And the way this project started is the clinical microbiology came to us and said, we have this outbreak and we don't understand it. And what they didn't understand is that they never had multi-drug resistant Acinetobacter in the hospital before, but their standard molecular typing approaches were telling them they had 3 different strain types. So what do you make of that, right? Like, they don't understand. So the question was, were they really unlucky and had these 3 independent strains come to the hospital at the same time, which is plausible. This is becoming a more prevalent organism. Or were their standard molecular typing approaches misleading them? So what we did is we performed whole genome sequencing on each of the major strain types, and we were really able to understand what happened. We're able to trace 2 of the strains back to a single patient who likely brought those strains from another hospital. We're able to trace the 3rd strain back to a non-multi-drug resistant strain present in the hospital a year before the outbreak that in the intervening year had evolved multi-drug resistant and gone on to cause an outbreak. And this was really quite amazing to us and to our clinical microbiology colleagues that this technology, which at the time, and this is 2009, 2010, really hadn't been applied in this realm, and was giving them information that was leaps and bounds beyond any sort of molecular typing or any other typing methodology that they had available to them. And this is really exciting to us. The second work, which I want to briefly mention, was something I did more recently with a really talented postdoc student, Joy Yang. And with Joy, what we did is we developed a computational pipeline to automatically design multiplex PCR typing assays from genomic data. So the way this will work is you input a bunch of genome sequences for whatever organism you're interested in, and what will happen is the optimal set of primer pairs will be selected to distinguish all the input strains in a multiplex fashion. And I think that the significance of this work and work like it is that the application of genomics to track infections in hospitals doesn't have to be limited in the short term to those hospitals that can afford to put sequences in the clinical microbiology lab. and that there's a glut of genomic data, especially for bacteria, that could be mined in creative ways to design clinical assays that could be implemented right now in a cost-effective manner. Okay, so with that, I'll move on to the two major stories. Yes.
Just by putting sequencing in classical microbiology labs, I mean,
Yeah.
they use the same infrastructure to actually sweep through the hospital and do the infection control.
Yeah. So
I mean,
I think the infrastructure is there or can be there to go into clinical micro labs. And what I expect will happen is that Illumina will team up with some more traditional clinical micro company, which is already happening. They announced a deal in the past few weeks with BioRimio, and working together, they'll have an all-in-one infrastructure that can be implemented at labs. But I think in the short term, that's going to be limited to larger academic institutions that are a little more flush. And there are a lot of smaller institutions, other sorts of healthcare settings besides acute care hospitals, like nursing homes, like long-term acute care hospitals that need more financially realistic methodologies. And exploiting genomic data to design these sort of cheap methodologies, I think, is a way to go in the future, at least for the short term. Yes. Yeah, go ahead.
Someone I've worked with at McGill.
Okay.
He does. They do this 24-hour turnaround.
Yeah.
They will do whole genome sequencing on any pathogen for the Canadian Public Health Service.
Yeah.
And they guarantee 24-hour turnaround.
Yeah.
And I mean, it's pretty amazing. I don't know if you know his work, but he's been doing this since about 2005.
It's not 24 hours.
It's mostly on things that aren't really known, where people don't really know the cause of the illness.
Yeah.
He calls them something like severe. He has some acronym.
Yeah.
For them, almost like SAD or SAP. Do you know what I'm talking about? Severe adverse pathogens or something like that? Yeah,
so there's been some studies that have got a lot of press, like New England Journal of Medicine level studies, where there's been an unknown infectious agent, and they'll directly sequence clinical samples and identify that agent.
Yeah, that's what he does. I know it's different than what you're doing.
Yeah.
His work is pretty phenomenal. I mean,
and I think, in the studies that I'm going to talk about, time was a limitation, and the quickest we could really get a sequence from a clinical isolate was 72 hours. Which is fast, but it's not real-time fast, where doctors really need information within hours for it to be practically useful. But I think that hurdle will be overcome sooner than later. So the first story I want to talk to you about is the application of genomics to study the evolution of resistance to a last-line drug in Acinetobacter. And the second story will be using whole genome sequencing to track this outbreak of carbapenem-resistant Klebsiella at the NIH Clinical Center. So the Acinetobacter story was published last year in Genome Research. So this is data from the same study looking at antibiotic resistance in ICUs across the United States, except now I'm just showing you for Acinetobacter, and I broke it down to individual drugs. And I really just want to emphasize that the trend is the same. Again, this is just a 10-year period. You're seeing increasing levels of resistance to every single drug, except for this blip, which I think is an artifact in the study. And by the end of this study, the only drug with a reasonable rate of susceptibility are what's called carbapenems. And as you might expect, these became the go-to drug to treat multi-drug resistant Acinetobacter. And as you also might expect, if you took this out to 2013, you'd see a sharp increase in carbapenem resistance. So again, we're left in a place where there's really no effective drugs to treat this organism and multi-drug resistant strains of this organism. So the lack of available drugs led doctors to reach back into the past. Yes, go ahead. There's definitely an accumulation of resistances where there's an obviously selective benefit to accumulate as many resistances as possible, and that's what we've seen. Yeah, I mean, part of it was the order in which they were developed. That this is happening over several decades, the accumulation of these resistances. Yeah, but Yeah, I mean, what's typically done is, like I said, a doctor won't know immediately what the susceptibility is. It takes 48 hours, so they have to treat the first 40 hours based on assumption. And for the best interest of the patient, they're going to use the most broad spectrum drug that they can use. And then if they can, they'll tone it down after that. And that's part of the problem. And that's why we need to be able to know with certainty what the resistance is in real time. To use the most specific drug you can use and save the higher level drugs for these really multi-drug resistant strains. That would be the optimal thing to do. So the lack of availability of standard drugs has led doctors to reach back into the past and go to this drug Colistin, which was used in the 70s and then discontinued because of associated neurotoxicity and nephrotoxicity. Now there are no other options, so you're willing to deal with these side effects to treat these severely resistant infections. Colistin is a cationic peptide antibiotic, so the way it works is it's positively charged. It interacts with a negatively charged bacterial membrane, gets into the periplasmic space, and ultimately causes membrane disruption and lysis. So now that Colistin use has begun to increase, we of course have seen resistance begun to emerge in hospitals. And whole genome sequencing clinical isolates has revealed this PMR operon is really important in resistance. So we see mutations occurring in this region of the genome in resistant isolates. And when resistance evolves, it typically happens as follows. So it's a 3-gene operon, and we'll typically have a mutation in PMRB, which is signal transduction kinase. This is now a single point mutation, so it's a very easy resistance to get. And it will hyperactivate the kinase, leading to hyperphosphorylation of a PMR8 transcription factor. It's autoregulatory, so this drives transcription of the whole circuit, including the 3rd gene, which is really the payload of resistance. It encodes an ethanolamine transferase, which transfers ethanolamine moieties to the bacterial membrane, decreases the negative charge, and inhibits interaction with the drug and provides resistance. So the question is, now that we've seen resistance begin to emerge in hospitals, is widespread Colistin resistance a foregone conclusion, which again would mean essentially the end of the antibiotic era for multi-drug resistant Acinetobacter and other very resistant gram-negatives. And kind of the obvious answer is of course widespread resistance is going to occur. It can be acquired really easily, a single point mutation, and why should Colistin be different than all the other drugs that we've seen fall by the wayside? But there's an alternative camp who's kind of holding out hope that Colistin might be special. And they hold their hat on 2 things. The first is that Colistin is part of a larger class of drug called antimicrobial peptides, and members of this class of drugs are also part of the innate human immune system. People have observed that they've maintained their efficacy against potential bacterial invaders over millions of years, suggesting that there might be something fundamental about this class of drug and the associated mechanism of resistance that might make stable resistance really difficult to evolve, at least for certain organisms. And that's a little hand-wavy, but kind of hard experimental evidence that this might be true comes from both in vitro and animal studies showing that Acinetobacter acquires a really significant fitness cost when it becomes resistant to this drug. So becoming resistant to Colistin results in it growing slower and being less virulent in animal models. So that there's something at play here. And I just want to say a little bit more about this idea of a fitness cost, because it's really critical to understanding the rest of the talk. So resistance to many antibiotics is associated with a cost. And the rationale makes a lot of sense. So antibiotics are inherently targeting essential bacterial machinery, which has been optimized over billions of years. So mutating this machinery in order to evade action of the drug comes at a cost, right? You're going to grow a little bit slower. But that being said, given that most drugs come at a cost, we've seen stable resistance evolve to other drugs. So what's happened? So there's 2 ways around this. The first is to attain, the jackpot, the low-cost mutation, which provides resistance with a minimal fitness cost. And interestingly, if you look at clinical isolates that we're isolating from infections in hospitals, they're enriched for these low-cost mutations, kind of further demonstrating the significance of this idea of a fitness cost. That these are the ones that are the most fit, and these are the ones that are spreading through our hospitals. An alternative route to stabilizing resistance is to get a secondary mutation on top of the resistance mutation that alleviates the fitness cost. So the big question is, are either of these possibilities available for Colistin resistance in Acinetobacter? So that brings us to the current study. This is what we're interested in, understanding whether there's low cost or secondary mutations that can alleviate the fitness cost of Colistin resistance in Acinetobacter. So the patient population are 40 patients at the NIH Clinical Center who have multi-drug resistant Acinetobacter infections that were treated with Colistin. Of these 40 patients, we see resistance evolve in 4 patients. And I think really importantly, it appears that resistance evolved de novo in each of these patients because their initial isolate was susceptible to the drug. So 4 to 40, we see resistance evolve. Again, a high probability resistance. And also importantly, after resistant isolates were taken from these patients, Colistin treatment was stopped because of the associated toxicity of the drug. So this is kind of where we're starting. We're ready to sequence these resistant genomes and look for low-cost mutations. And we made an observation that was really interesting and kind of made us rethink how we were approaching the study. And this is what the observation is. It's that soon after the drug was taken away in these 4 patients, resistance appeared to disappear. So this is one patient. Here's the x-axis is time. The y-axis is the level of Colistin resistance. This dotted line is the clinical threshold for what you would call a resistant isolate. And the gray box is the Colistin treatment window. So we see the initial isolate sensitive, treatment of Colistin starts. We're getting resistant isolates in this patient, treatment stops, and now we're getting susceptible isolates. And this is like a week after the drug was taken away. And we see the same phenomenon in all 4 patients. And what this really is indicating is now an in vivo manifestation of the fitness cost, right? As soon as the selective pressure of the drug goes away, susceptible isolates are quickly outcompeting resistant ones and retaking the infection. And this is pretty amazing to us. This is not something that's ever been seen with any other organism-drug combination. And what it implies is, one, we don't have to look for low-cost mutations. They clearly don't exist because susceptible isolates are quickly outcompeting these resistant ones. But what it suggests that might be interesting is that what's happening here to cause them to lose resistance, and what are the implications of this on whether Colistin resistance will become widespread? And that really became the critical question that we were interested in answering. Yes.
Who are the patients with respect to one another? I mean, how does this move around?
Yeah, so it depends on the pathogen. Acinetobacter is kind of a very difficult one to control because it could survive in the GI tract, it can survive on your skin, and it can survive on inanimate objects for long periods of time. So there's many different ways where it could spread. In the literature, there's outbreaks associated with contaminated ventilators, with humidifiers, which is kind of frightening, with anything you could think of.
Yeah, we were talking about this, just think of the hospital in the future.
Yeah.
You go into an ICU room, or any hospital room, it's got all these little training.
Yeah.
I mean, we've got to redo the whole thing.
Yeah, and especially with Acinetobacter and another organism, enterococcus. Yeah. So the study design was extremely simple. We performed whole genome sequencing on longitudinal isolates from each patient, including the initial susceptible isolate, at least one resistant isolate, and the post-treatment susceptible isolate. And the first thing we find is kind of just confirming what we knew. We looked at the resistant isolates, and they all had mutations in PMRB, which is what had been found in previous studies. And just to confirm that things made sense, we also measured the expression levels of the PMR locus, and they were constitutively activated in all resistant isolates. So this is, again, just kind of a sanity check and confirming that things make sense. Now, the more interesting question about how is resistance being lost in these different patients? And a priori, there's 3 possibilities. The first is reversion. You could have a mutation conferring resistance and a secondary mutation going back to the ancestral genotype. You could have re-emergence, where you have a mutation causing resistance, and then once the drug is gone, the ancestral susceptible population comes and retakes the infection. And you can imagine this happening if you have an anatomical site where cytotoxic drug concentrations aren't reaching. Then once the drug is gone, it can kind of rediffuse throughout the patient. Or you could have a dormant population of bacteria that, once the drug is gone, again, could come and reseed the infection. The final possibility is compensation, where you could have a mutation conferring resistance and a secondary mutation ablating resistance. And before moving on, I just really want to emphasize that this third possibility is fundamentally different than the other 2, in the sense that what you end up with in the end is different than what you started with. And it's different in a really important way, where there's been a secondary mutation that has essentially deactivated the resistance locus to this drug and could have implications on the capacity for the future evolution of resistance. Yes, go ahead.
Is this a total extension of a single resistant mutation, or is it like a quasi-species with like multiple mutations and different?
Yeah, it probably depends on the patient. And what really drives resistance are pre-existing mutations. So it depends on how large the bacterial population in the patient is at the beginning. but it's not like a virus where every single possible mutation of the genome exists at all times. It's lower mutation rate and lower population levels. But that's a really good question. And thinking about populations is kind of where this field is going. But we didn't have the samples to do that here. So what do we see in our 4 patients? Well, in the first patient, we see a compensatory mutation. So to kind of step back for a second, looking longitudinally in these 4 patients, we see between 2 and 10 mutations occurring over time. I'm only going to show you mutations in the PMR just for simplicity and because you can really get the take-home message just by looking at mutations at this locus. So the resistance isolate, we see the PMRB mutation, as I mentioned. And just to refresh your memory, this is activating the signal transduction kinase, hyperphosphorylated transcription factor, driving transcription from the locus, and providing resistance. Now, in the post-treatment susceptible isolate, we still have this PMRB mutation. So presumably, this kinase is still hyperactivated. But now we have a mutation in PMRA, the transcription factor. And if you look closely at where this mutation is, it's right smack in the middle of the DNA binding domain of the transcription factor. So what appears to have happened is that in order to lose resistance, it's essentially mutated and destroyed the transcription factor that's critical to resistance. Which again is just like kind of dumbfounding to us that accidentally by giving a patient a drug and taking it away, we've selected for the organism to destroy this critical locus. And it made us wonder, what would have happened if we brought the drug back to this patient? Would it be harder to evolve resistance again? And the idea is that initially it was very easy to mutate PMRB to evolve resistance. There's lots of different ways you can change amino acid sequence and confer resistance. And we see that based on the fact that we see different mutations in each patient. But once you have the PMRA mutation at the DNA binding domain, it can be really hard to undo. You might need a true reversion to bring resistance back. So this is a retrospective study, so we couldn't do this obviously in the patient. So we had the isolate, so we wanted to see, is it harder for this post-treatment susceptible isolate to become resistant? So we did a very simple experiment. We measured the rates of Colistin resistance mutations in vitro for the pre-treatment susceptible isolate and the one with the PMRA mutation. And the way the experiment works is you start with a small bacterial population. You grow it in non-selective media just to accumulate mutational heterogeneity. And you plate the population on a Colistin plate, a non-selective plate, and you look at the frequency of mutations. So I did this experiment myself, which is one of the first actual experiments I did with my hands. And I kept getting different results every single time. And my first intuition is that I'm a novice, so this must be experimental incompetence. But ultimately, I learned that it wasn't experimental incompetence, that there was a Nobel Prize-winning reason why I kept getting different results. And it's as follows. Maybe you're familiar with this experiment. So the number of Colistin-resistant colonies you get depends on when the mutation occurred in the outgrowth. So in each of these 3 cases, there was a single resistance mutation that occurred, but the number of colonies you see depends on whether it happened early or late in the outgrowth, right? So the way around this is to do the experiment many times, build a distribution of the number of Colistin-resistant colonies, and infer the most likely associated mutation rate to explain the distribution. And this is what Lurie and Delbruck did in their Nobel Prize-winning experiment. This is what I thankfully discovered that I should do before I submitted this paper, so I didn't like a total jackass. And this is what I did, and here's the results. So we see 2 distributions. This is the pre-treatment susceptible isolate. This is the post-treatment susceptible isolate with the PMRA mutation. The distributions are clearly very different. The inferred mutation rates are different, and it's highly significant. So again, the critical take-home message to me is that phenotypically, these isolates are identical. They're susceptible to the drug, but they differ in their capacity to evolve resistance. But there'd be no way for a doctor to know that without having the genetic information and without having this nuanced understanding of the mechanism of resistance to be able to predict that this PMRA mutation should inhibit the capacity to evolve resistance. So that's just one patient. What about the other 3? So patient 2 and 3 both have PMRB mutations in the resistant isolates, and they're both gone in their susceptible isolates. So we think it's re-emergence. In principle, this could be explained by a reversion, but based on the other mutations that I'm not showing you, it's likely a re-emergence, although, again, it doesn't really matter. The key thing is that what you end up with is what you started with. 4th patient also looks like re-emergence. We sequenced 2 resistant isolates here. They have different PMRB mutations, likely this population effect. We're sampling different resistant isolates from the population. But regardless, both of them are gone after you pull the drug away. So at this point, things are actually looking pretty good for Colistin, right? We have 4 patients, admittedly a small number. But in 3 of the 4 patients, you're no worse off than he started with in terms of being able to bring back the drug and having it be efficacious again. In 1 case, you might actually be better than you started with, where the drug is more likely to not incur resistance. So unfortunately, the study didn't end on this optimistic point, and we continue to push on. And the reason we didn't stop here is because of an observation made in our clinical microbiology lab. So when a sample comes into clinical microbiology, say a sputum sample, they'll plate it out and they'll look at the colony morphologies, and if they're all the same, they'll take a single colony and they'll work it up and determine the susceptibilities, and that's kind of the end of the day. And that's what was done for all the samples in the study, except for samples taken from patient 4 while this patient was on the Colistin treatment. When they plated these samples out, they had 2 different colony morphologies. They had large colonies and small colonies. The small colonies, it turned out, are Colistin resistant, and these are the ones we sequenced that had these PMRB mutations. The large colonies, it turned out, are susceptible to Colistin, and we sequenced them, and they have the ancestral PMR genotype of patient 4's initial isolate, right? So an apparently susceptible genotype. So first of all, knowing that the Colistin resistance phenotype explains the different colony morphologies, right? This is another manifestation of the fitness cause. Resistant isolates grow slower, so they have smaller colonies. But from a clinical perspective, the key question is, why are we isolating Colistin susceptible isolates from a patient receiving the drug at an anatomical site where it appears that cytotoxic drug concentrations are reaching, based on the fact that we're isolating resistant isolates? And the concerning possibility was that maybe patient 4's initial isolate already had a low level of Colistin resistance that we weren't detecting, that was allowing it to stay in the population during the course of treatment. So to test this hypothesis, we went back to the genome sequences, and we took advantage of the fact that patient 2 and patient 4 were part of the same outbreak, so they had very closely related strains. And we asked, is there something special in the genome of patient 4's initial isolate that distinguishes it from patient 2's initial isolate that could explain this observation in clinical micro? So it turns out that there were 8 amino acid changing mutations differentiating these strains, and one of the 8's in PMRB, this critical gene for Colistin resistance. So this doesn't seem like a coincidence. We went and did the expression measurements, and in fact, patient 4's initial isolate has significantly higher PMR expression than patient 2's initial susceptible isolate. And I think also important to note, it has significantly less PMR expression than patient 2's resistant isolate. So it's somehow a mutation that confers an intermediate activation of this signal transduction cascade, an intermediate expression and an intermediate level of resistance that was totally missed by the clinical assay. And to kind of hammer it home and put the final nail in the coffin, we did a different Colistin resistant assay. So the clinical microlab, they're doing a plate-based assay, we did a broth-based assay, and using that, we found clinically significant levels of resistance in patient 4's initial isolate. So it had just been missed by the clinical microlab because this mutation provided a phenotype that was missed by their assay. So to summarize this part, first, I think the summary of the bad news is that we see a stable Colistin resistance mutation emerge undetected by clinical assay, and we went on to show that this resistant mutation was transmitted to other patients. So this is what you were concerned about, right? We're having now a stable mutation that's fit enough to actually be transmitted to other patients. And I put resistance in quotes here because it's not really a true resistance mutation, right? We see other PMRB mutations happen on top of this genetic background. What it really is a mutation that predicts the evolution of resistance. It provides kind of a low level of resistance that allows the bacteria to stick around during treatment, acquire better mutations, but then at the same time, come back and retake the population after the drug is gone. And kind of feeding off this same point, this could be a broad mutational mechanism that could allow for circumvention of high-cost resistance mutations. So, you know, again, you have this wimpy resistance mutation that provides just a little bit of resistance, but with very minimal fitness cost. You could stick around during the presence of the drug, which is inherently a temporary inconvenience. And when the drug is gone, you could come and retake the infection and be transmitted to other patients, which is what we observe. But I think there is some good news. And I think the first piece of good news is that genomic analysis revealed that with proper selective pressure, the probability of resistance evolution can be decreased. So here this was in a really trivial way. Accidentally, by giving a drug and pulling it away, we selected for isolates that had trouble evolving resistance to Colistin. But you can imagine doing this in more intelligent ways, where say you had combinations of drugs, where the first drug is given to decrease the probability of resistance to a second drug. And since this paper has been published, there has actually been publications that have come out showing that combinations of existing antibiotics have this property which they term collateral sensitivity, where resistance to one drug will select for susceptibility to another. And you can imagine both in long-term colonized patients using these sort of treatment strategies, and also within a hospital using these sort of strategies to use one drug for a period of time in a hospital, kind of wipe out resistance to the other drug, and cycle back and forth in an intelligent way like that. The second piece of good news, I think, is that genomics informed the current and future state of resistant strains. So we could look at this PMRA mutation and predict that resistance to colistin should be difficult to evolve. We could look at this PMRB mutation in patient 4 and say, you know, the clinical phenotype is telling us it's susceptible, but this looks like a resistance mutation. And I think what this speaks to is the need for genetic information to supplement phenotypic assays when doctors are deciding what drugs to use for patients, with the added benefit that this can be information that is given much quicker to a doctor. So I think the first story really sets up the second. So we're seeing that the final drugs that we have to treat these infections fall by the wayside. And again, now the onus is really on infection prevention in the short term. And what I'm going to talk about is the use of genome sequencing to try to understand how infections are spreading in hospitals, with the ultimate goal to understand to a point where we can implement infection control protocols that will really limit the spread of infections. So a little background on Klebsiella outbreaks. And they primarily occur among immunocompromised patients. And the way they're thought to spread is primarily patient to patient on the hands of healthcare workers and on medical devices. So unlike Acinetobacter, it's not very stable in the environment. It's not typically long-term colonizing rooms or other infrastructure. This is really short-term spread going from room to room. And what makes these outbreaks really difficult to control is that in addition to causing infections, Klebsiella could asymptomatically colonize patients' GI tracts. And what this does is it creates silent reservoirs for ongoing transmission, where unless you identify all these asymptomatically colonized patients, these outbreaks can really continue in perpetuity. So up to 2011, we were fortunate not to have any carbapenem-resistant Klebsiella pneumoniae, which is what we're talking about here, and carbapenem being the really last line drug for treating these infections, so they're very serious. And then we had our first patient who was transferred in from another hospital, and this patient was actually known to be colonized with this organism upon arrival. So to prevent an outbreak situation, this patient was immediately placed in contact isolation. And there were 2 exceptions to this patient being in isolation, which were 2 24-hour stints the patient spent in the ICU as part of their standard care. And to try to assure that no transmissions took place, both throat and groin surveillance cultures were done on every other patient in the ICU. They all came back negative, so it seemed that everything was fine. This patient ultimately left the hospital in mid-July, and there had been no other cases, so everything seemed good. 3 weeks after patient 1 left, a second patient came up positive, and then a third, fourth, fifth, and then the floodgates opened. And when all was said and done, a total of 18 patients had been affected, and 12 of those 18 patients ultimately succumbed to their infections, which is pretty standard for the mortality rates for this sort of organism in this really immunocompromised patient population. And infection control was left to wonder what had happened here. And despite following all the proper infection control procedures, this had happened. And to really understand what had gone wrong, there were 2 critical questions, I think. The first question is, what is the link between this first patient and the rest of the outbreak? Is it just a coincidence that this patient was here, or is there a causal link?
Where did they come from?
A New York hospital, which is where this organism is most prevalent in the country. And regardless of whether patient 1 is linked to the outbreak, the really striking observation is that once the second patient came up positive, things happened real fast. It was 1 a week for 4 months. So why did things happen so quickly here and not here if patient 1 was linked? So how could we begin to understand what had happened here and try to recreate this outbreak? So do you mean, do they do like surveillance culturing? I'm sorry, where did the question? Oh. So they didn't in the study. They're doing anonymous studies now to do that. They're all coming back negative. But there are like major ethical and legal ramifications to doing those sorts of things that I think have limited that from being done more broadly. And the limitation is, what if a nurse is colonized with this pan-resistant organism and they can't be decolonized easily? What do? Do you fire them? but it's not that easy, right? And that's the easy answer. I agree, but it's not a trivial decision to make. It is more complicated. But that being said, other places in the world are doing this, and there have been studies published out of the UK where they have done surveillance on healthcare workers. In a case of a neonatal MRSA outbreak, they traced it back to a healthcare worker, and they published it. So it's an open debate, and above my pay grade, unfortunately. Yes, go ahead. Yeah. So that's right on the head, and I think there's advantages to it in terms of resources, in terms of isolates, in terms of being a research environment, and facilitating this study when we did it. Yeah, it's a very resource-rich environment, which is one of the points I was alluding to earlier, that in the near term, it's unlikely that this is implemented in real time in all hospitals. It's just not practical. But I think the lessons that you can learn from doing this in large centers can be applied broadly. And I think that'll be a take-home message here, that what we learned here can be used elsewhere in more resource-limited settings. So what's traditionally done in outbreak investigations are molecular typing approaches other than genome sequencing. So for Klebsiella, that amounts to pulse field gel electrophoresis, where you take genomic DNA, you chop it up with a restriction enzyme, you run that on a gel, and you have like a molecular fingerprint, right? Rep-PCR, you do PCR in repetitive regions of the genome, and again, you put it on a gel and have a molecular fingerprint. It turns out when you apply these approaches to the 18 isolates from the outbreak, they gave identical banding patterns. So there was really no information distinguishing the isolates. But you might say, well, at least you could link patient 1 to the outbreak because it's got the same banding pattern. But it turns out that that's not even true, because carbapenem-resistant Klebsiella is highly clonal in the US hospital system. And having an identical banding pattern doesn't distinguish a transmission within the outbreak from an independent introduction. So there's really zero information here. This is totally worthless. And you can see, to emphasize that, I put a really low quality graphic. So molecular typing doesn't work. What about gumshoe epidemiology, kind of the standard of care? Here I'm showing you traditional epidemiological data. This is called a patient trace. On x-axis we have time, y-axis we have our different patients, and the different colors represent different wards where the patients were during the course of the outbreak. So we think that these outbreaks are spreading patient to patient via healthcare workers' hands and medical devices. So it stands to reason that transmission should be happening between patients who are in the same ward at the same time during a period when we think transmissions are happening, right? So what if we tried to recreate the outbreak using this sort of assumption? Can we recreate the path of transmissions? So this is what that network looks like. And you can see it's not a pretty one. And the reason it's not a pretty one is that this is primarily an ICU outbreak. So everyone overlapped with everyone. So again, that there's no information here. And this is what ultimately led us to apply whole genome sequencing. And again, the idea really is not very complicated. We have our outbreak, we have our isolates, we identify the mutations, and we use really standard phylogenetic approaches to infer the ancestry of the isolates, and then in turn infer a putative path of transmission. And we were not the first ones to apply this idea. And in fact, in the years leading up to this study, there were several really high-profile papers applying whole genome sequencing to study the worldwide dissemination of like the who's who of infectious agents. So this is a really cool figure from a paper looking at the worldwide dissemination of Vibrio cholera. So what they did is they took isolates of Vibrio from all over the world, isolated over the past several decades, performed whole genome sequencing, did a slightly more sophisticated phylogenetic analysis, and it came up with this map. And among the interesting things you can infer from this map are that all 3 pandemic waves of cholera emerged from the same part of the world, from Bangladesh. And in the kind of current events of the time, they're able to trace the origin of the cholera strain that caused the Haitian outbreak after the 2010 earthquake. And before the sequencing was done, it was assumed that the source was South America or Latin America for obvious geographical proximity. But genome sequencing pointed to the fact that it was from the same part of the world, Southeast Asia. And subsequent epidemiological investigations supported this and ultimately linked the strain to Nepalese UN peacekeepers. So now we're seeing genomic data driving epidemiological investigations, which is really what we want to do here. And ideally what we want is to take this sort of map and put it on our hospital. So let's do that. But can we is the question. So this sort of study is relying on variation that's accumulating over decades. We're trying to infer events that are happening over days and weeks. And a priori, it's not obvious whether Klebsiella or any other hospital pathogen is mutating fast enough to give us the variation that's sufficient to make these sorts of inferences. Potentially, we could sequence our 18 isolates from this 4-month outbreak, and they could all be identical, and we'd be no better off than pulse field. So that was kind of a risk we were taking. And our first indication that this wasn't the case is looking just at isolates from patient 1 from different anatomical sites over time. We see variants. So here I'm showing you different isolates on the y-axis and variable positions on the x-axis. So there's 6 million base pairs in the Klebsiella genome, and of those 6 million base pairs, there's 7 positions where they're not identical. So these are very, very closely related, but there are differences. So this is encouraging. And kind of at the risk of reading too much into the data, there also seems to be an anatomical structure to mutational patterns where all the isolates from the urine are identical and have different mutational patterns from the BL and the groin, which in turn has a different mutational pattern than the throat. But that's neither here or there. The critical thing is that we're seeing variation, which was encouraging to us. Next, we look at the full set of isolates. So again, the isolates are on the y-axis, variable positions on the x-axis, and now we're seeing roughly 40 variable positions out of 6 million. So still very closely related, but differences. Roughly half of the variants are unique to a single isolate, so they're not useful for a phylogenetic analysis. The other half are shared among different sets of patients, and there's a clear structure there, which you could see based on the way I clustered it. And potentially, we could use these patterns of shared variants to infer transmissions in the outbreak. So this is great, but can we actually learn anything that's clinically interesting? So the first thing that's very clear is that patient 1 is part of this outbreak. We see a single nucleotide variant distinguishing patient 1's throat isolate from the isolates from patients 3 and 5. And in the context of the rest of the variation that we're seeing evolving during the course of this outbreak, I mean, that's as clear as you can get that this patient is part of this outbreak. And I think even more interesting than linking the patient was additional evidence that there were multiple transmissions stemming from this patient. So you see two major clusters here. This first patient with patients 2, 3, and 5 share the mutational patterns from patient 1's throat isolate. Everyone else shares the mutational patterns from patient 1's BEL and groin isolates. And because we know that patient 1 started the outbreak, we know these variants evolved in patient 1, we have a flow of directionality from patient 1 to the other patients. So at least 2 transmissions, and actually I would hypothesize more than 2 transmissions based on the genomic data. And this is really critical. If you remember back, patient 1 was discharged 3 weeks before anyone had any idea of an outbreak. So what this is telling us is that the reason that things got out of control so fast here is not because there were a lot of transmissions after this point, but there were a lot of transmissions that went undetected early on. Patients were asymptomatically colonized, and by the time anyone knew there was an outbreak, it was probably all over the place. What this also tells us is where and when these transmissions likely occurred, right? So, and the obvious culprit is these 2 24-hour stints that this patient spent in the ICU when groin and throat surveillance cultures were done to try to rule out transmissions. And ultimately, the hospital switched to rectal surveillance cultures, which are much more sensitive for detecting asymptomatically colonized patients because it's colonizing the GI tract. And this is really critical to ultimately stemming the tide of the outbreak. So the really important thing is that now we're providing hard evidence for where and when things went wrong, what could be changed to prevent this from happening in the future, and explaining kind of this broad epidemiological phenomenon that would have really been unexplainable through any other way. So that was kind of nice, and cherry picking specific questions that we're interested in, but obviously ultimately what we want to do is infer this sort of transmission graph. So I implemented a maximum parsimony algorithm on a graph, which is called Edmonds algorithm, to find the minimum spanning tree on a directed graph. And this is what it gives you. And among the interesting things, again, now it's not 1, 2, but 3 transmissions stemming from patient 1, which makes sense when you look at the genomic data. And I just want to go through actually one additional predicted transmission just to kind of to emphasize a point. And the predicted transmission is going 1 to 3 to 2. So first of all, the numbers indicate when patients came up positive. So the reason you could even have an out-of-order transmission is because the capacity for asymptomatic colonization dictates that when a patient came up positive isn't necessarily the order in which they acquired it, right? So this is possible, but does it make sense? So if you look at the genome sequences, the reason that that transmission is being predicted is because of a single mutation in the genome of the isolate from patient 2, which is not in the isolates from patients 1 and 3, with the presumption being that the mutation happened after the transmission from 3 to 2. So that's great, but does it make sense if you go back and look at the epidemiology? And in fact, it does. We see that patient 1 overlaps with patient 3 in both 24-hour stints in the ICU. Patient 2 never overlapped with patient 1, but later on overlaps with patient 3 in the ICU. So now we're seeing that the genomic data and epidemiological data are telling us the same thing, and this is really where the power of this approach is going to come. The genomic data can't give you everything, and epidemiological data is really unvalidated. By putting them together, you really gain a complete understanding of what's going on here. So what did the study yield? The first thing I think it clearly yielded is that genomics provides unprecedented resolution in tracking the spread of hospital pathogens. We provided hard evidence that Klebsiella outbreaks can spread undetected for extended periods of time, and we provided evidence for where and when breaches in infection control take place. What did the study not yield? So I kind of want to pump the brakes a little bit and not leave you thinking that this is the cure-all. So it didn't provide an infallible map of all transmissions that happened throughout the outbreak. And there was actually a good deal of luck that this was so successful. So the critical piece of luck is that what we were interested in was these transmissions really early in the outbreak. And because we knew that patient 1 started the outbreak once we were able to link them genomically, we had a kind of flow of directionality for the transmission. We knew everything originated with patient 1. If we didn't know that, things become much trickier. So let's look at another example. We have patients 12 and 13, which have an identical mutational profile, and only 2 patients in the whole outbreak that have their mutational profile. So it's reasonable to infer that there's likely a transmission between these 2 patients. But was it 12 to 13 or 13 to 12? Totally non-obvious, and there's really no information here to give you the answer to that. Things can become much worse. So if you look at this Edmonds algorithm transmission graph, it looks like patient 6 really is a big problem here, right? It looks like a super spreader, but that's really not the case. So if you look at the genomic data, in the middle of the outbreak, there's very little variation accumulating, and this is just one of many possibilities to explain the genomic data. So really what I'm getting at is while they are evolving fast enough to give us some insight, there's not sufficient mutations to completely parse everything out. But there's clearly information here. So how do we strike the balance and really use this information to its maximal effect? And I think what has to be done is to really use the genomic data to rule out transmissions that couldn't have happened, look at the reduced set of possible transmissions, and hopefully find enrichments for clinically interesting things in this reduced set of possible transmissions. And I want to spend 2 slides just telling you about one example of how we're trying to do that right now. So this is now looking at another organism, vancomycin-resistant enterococcus, and we performed whole genome sequencing on roughly 60 isolates at the NIH Clinical Center from a 4-year period. And what we attempted to do is, again, use genomic data to limit transmissions, overlay epidemiological data about where patients were in the hospital, again, to reduce transmissions, and look at this reduced set to see if we could learn something about the epidemiology of this organism. So in the past several years, there's been a lot of what's called a case control study trying to understand factors that put patients at risk for acquiring VRE, or vancomycin-resistant enterococcus. So what you'll typically do is have patients that have VRE, have patients that don't, and see what's happening in the 14 days or 30 days prior to getting the infection that seems to be enriching patients for putting patients at risk. But there's, I think, an underlying flaw in these studies. What they're assuming that they're doing is, you know when a patient came up positive, so you look in the window right before they came up positive and say, okay, what's happening here? But there's another possibility, which is this asymptomatic colonization. So just because a patient came up positive here doesn't mean they acquired it here. They could have been colonized for a very long period of time, have acquired it long ago, and then something happened here where you're able to detect it, or maybe you just didn't look before, right? And it's really important to distinguish these 2 populations, because what you're finding here are factors that put patients at risk for acquiring the organism. What you're finding here are factors that put patients at risk for testing positive, which is equally valuable to know, because you could target these patients for surveillance culturing when you find these factors. So we wanted to use genomics and epidemiology to split these groups up. And the way we did that is we put patients in this group if they have a genetically plausible transmission scenario from another patient with whom they overlapped in the hospital in the 14 days prior to coming up positive. Those are patients who are recent acquisitions. If you don't have that scenario, we assume that you're long-term colonized. So we split patients up like that, and we asked, okay, when we split patients up like this, are there differences in the antibiotics that these patients are receiving prior to coming up positive? And in fact, to kind of my surprise, because this is kind of complicated data set and clinical data, that we did find something interesting. So if you looked at the long-term colonized patients, there's a single antibiotic that they're enriched in, oral vancomycin. So remember, these are vancomycin-resistant enterococcus. So patients receive vancomycin, and you're essentially wiping out vancomycin-susceptible organism, enriching for vancomycin-resistant organisms, and that makes sense that you'd be able to detect asymptomatic colonized patients when they're receiving oral vancomycin. So this is nice. It actually makes sense, which doesn't often happen with clinical research. Ceftazidime is highly enriched for patients that recently acquired VRE, and this doesn't make immediate sense to us. And what I hypothesize is going on is there's actually a hidden confounding factor that patients that are receiving prophylactic ceftazidime are enriched in some sort of underlying condition, and we're bringing in additional clinical data to try to identify this potential confounder. But I think it's pretty clear that these patients have been split up in a meaningful way, and this is really exciting to us. And I think this is kind of where the field is going now, trying to use genomic data to split patient populations up in different ways to understand possible transmission and look for clinical enrichments. And this is the sort of information that can really be used in hospitals to change clinical practice, hopefully for the better. So overall conclusions, we see that genome sequencing can inform transmission and evolution of hospital pathogens. And I think that the writing's on the wall that as the speed increases and the cost decreases, it's going to become feasible to incorporate this in standard clinical microbiology workflows. And with real-time insights into transmission and resistance, patient treatment can be optimized. So just to thank everyone involved in this work. So Julie Segre was my postdoctoral mentor and was really fantastic. I couldn't have asked for anything more from a mentor, and I had obviously a very productive and exciting postdoc, largely thanks to her. Clinical Center Epidemiology, Tara Palmore and David Henderson are the two head epidemiologists, and I really want to thank them because they're really doing the important work, right? They're really managing the outbreak and dealing with these patients and trying to prevent infections from spreading. But at the same time, I think they recognize the power and the transformative nature of genomics and still participated in these research studies to try to further them along and take full advantage of them. Robin Odom and Heather Hughes are both helping me out with the VRE work. These are clinical microbiologists who, in addition to providing endless insight, also let me do experiments in their lab as a computational person, which is very bold. It's our sequencing center and the Pratt Fellowship funded my postdoc, and NIH, and my first shout out to Michigan, which is exciting. And thank you so much for coming and listening.
So thanks, Evan. That was really fascinating work. I guess I want to ask the first couple questions here. So you kind of inferred this towards the end here on the slides, but on your directed graph, did you ever go back and incorporate what rooms individual was and the overlapping the kind of as tiebreakers between the transmissions?
Yeah, that's exactly what we did.
Did it make sense? I mean, were you able to resolve that, or?
Yeah, I think what was neat about that was seeing what didn't make sense, seeing what couldn't still be explained when incorporating that, and then inferring that there must have been a more complicated epidemiological scenario that kind of is counter to how we're assuming that transmissions are happening. So when genetics is truly telling you something that we have no epidemiological understanding, we went back and looked at those specific transmissions in particular and were able to learn interesting things.
Cool. And my second question, when I was looking at that graph, and I realize there's probably many factors, age, immune compromise, whatnot. But I was just thinking in terms of survival, if you were to overlay that on that graph, if there were any particular mutations that maybe made the bacteria even more resistant or more pathogenic, or
yeah. So obviously for the study, we don't care about phenotype. It's all there's markers, but there are definitely antibiotic resistance mutations that we see in the course of the outbreak, and that's just obvious from the phenotype. Where when the strain came into the clinical center, there were 3 drugs it was susceptible to: gentamicin, tigecycline, and colistin, which I mentioned earlier. During the course of the outbreak, resistance evolved to all 3 of those drugs, essentially again leading to pan-drug resistance, and those mutations are in those genomes. Other than resistance, it's really difficult to infer like changes in virulence or things that could affect patient outcome, but you would expect that over the long term those mutations are accumulating.
Other questions?
Yes.
I was wondering, the strains that you worked with, did you see any integron genes in them?
There, ooh, I don't know. There's definitely transposable elements. There weren't transposable elements movements during the course of the outbreak. There were transposable element movements in the Acinetobacter isolates over time in patients where in one of the patients that had a PMRB mutation, we also saw an insertion sequence jump right upstream of the PMR operon to probably further amplify expression from the locus, but there's definitely multiple mechanisms of evolution other than single nucleotide variants, so that's a really good observation.
Was the infection isolated? Was it in the ICU? I mean, what did they do? Did they just completely sterilize it?
Yeah, so
How did they get it out?
yeah, so they essentially created a cohort unit, so a unit that was specific for Klebsiella colonized patients or anyone who had been colonized, and they resided there for the duration of the outbreak, and then they would periodically do full hospital rectal surveillance culturing to identify any potentially silently colonized patients, and that was really the key to finding these silently colonized patients, so you can ensure that it was out of the hospital.
How many patients were involved? You know, how many patients are in the clinical center?
I would say, but like around 200. Yeah. So it was no small task. And again, I think a point that was raised earlier, there are resources there that aren't necessarily available. So this was something that Tara, our epidemiologist, decided that needed to be done. It was done. And in terms of bringing other molecular assays into the clinical micro lab, they can just do it. That sort of thing isn't necessarily trivial to do anywhere.
It's an international problem. It's growing worse. There was a White House level paperwork, you know, presidential directive after the PCAS study.
Yeah.
Mike Stebbins, who's my friend, was honcho of that.
Yeah.
Talking to him about a couple weeks ago about it. You know, our biggest concern is the hospital right over here. Is you know, a big infection within the hospital is one hospital away. Detroit Medical Center, Detroit Receiving Hospital, Henry Ford Hospital.
Yeah, and I'm engaging in some collaborations now with people who are interested in long-term care facilities. So these are facilities that have very sick patients. They're on ventilators, but and they typically go back and forth between hospitals. So there's really good infection control in hospitals relative to these places. And the idea is that these long-term care facilities could be kind of the epicenters that are seeding infections in hospitals. And there's been some really crazy papers showing that the colonization rate in some of these long-term care facilities is upwards of 30 and 40% with KPC Klebsiella. So like shocking. And to really get a control of this problem requires you know, the entire healthcare system to be explored, not just acute care hospitals.
All right. Well,
Great. Thank you so much.
Thank Evan again.
I got a picture of you